Good afternoon. Welcome to our session on gene editing, a strategy for the future. I'm Kevin Flanagan from Nationwide Children's Hospital, and I'm the moderator for today. We have terrific speakers, a terrific panel today. Dr. Melissa Spencer from UCLA, Dongshen Duan from University of Missouri, and Jeff Chamberlain from University of Washington, who are all thought leaders and, as you'll see, have great experimental data on gene editing. I'm going to take the moderator's prerogative, and I'm sorry, we've left our little block in there. We didn't take it out. Moderator's prerogative to show a couple of slides, just a couple of thoughts about gene editing before we start the panel. And I wanted to just point out, I don't know if you remember, I remember the first week of 2016, when this paper had appeared December 31st in the New York Times. There aren't many things in the field of muscular dystrophy where the New York Times has an article and everyone comes in the very next week and saying, all right, when do I get this? I'm not sure what they're doing. I think pulling down our red corners on that program. So well, let me just to continue for a minute. I'm going to give a very, very one-slide overview, a discussion of the technology itself. You'll hear a great explanation in a very nuts and bolts way, a very basic way from Dr. Spencer. And I want to talk a little bit and frame our discussion in really what it means for parents who are thinking about these therapies, when we'll have a chance to get to those kind of therapies as well. I think one of the challenges we have is the expectations versus the reality of gene editing, the challenges of delivering all of this, a gene editing approach to muscle, which is about 40% of the muscle mass, of their body mass. Other challenges that we have include how do we deliver it? What do we do with this? Oh, we'll continue here. I was winging it there for a minute, you forgive me. So that New York Times article referred actually to 3 papers, not the very first papers on Duchenne, muscular dystrophy, and genome editing, but 3 high-profile papers in science. And you know, from this day forward, parents have come in asking us, when are we going to be able to reach, when are we able to get CRISPR? I'm just going to use a brief slide to discuss terminology, and the CRISPR, some people ask sometimes, really stands for clustered regularly interspaced short palindromic repeats, which doesn't mean anything except to know that that's how they were discovered. These patterns of repeats were discovered in bacterial genomes. This is really discovered as part of the bacterial immune responses to protect against viruses. And Cas are CRISPR-associated system genes, nucleases, things that cut DNA. And nuclease can be targeted to specific regions of the genome by a guide RNA, and then the cellular mechanisms that repair nicked or cut DNA, those cellular mechanisms then, the attempt is to harness those, to use those therapeutically so that repair incorporates repair of the gene. That's the basic idea, and Dr. Spencer has a much more a clear description of it there. I just want to emphasize these challenges before we get to therapies. Targeting, I mentioned, how do we get CRISPR into all of the muscle? How do we get it to the places we have to be? How are we going to deliver it? You'll hear some about AAV gene therapy. We've heard a lot about AAV gene therapy delivery here, but CRISPR presents its own challenges with gene therapy. Can we deliver these proteins different ways? For example, putting them on nanoparticles. Even there's recent papers on using gold nanoparticles for it. There's problems, there's challenges, I should say, with the relatively low efficiency of gene correction. And then the big problem about when you break DNA, when you cut DNA or nick DNA, do you do damage elsewhere? All of these are significant challenges to it, not insurmountable challenges, but ones that we have to be aware of. So it's important to know when you come to clinic, you know, the question, when do I get CRISPR? CRISPR is not a single treatment. It's a platform for therapeutic approaches, and it's a platform that holds enormous therapeutic promise. Certainly, here's just 4 of the companies that have claimed CRISPR programs directed toward DMD. So there's people working very hard on this. You should know that somatic gene editing still faces a lot of hurdles for efficacy, safety, and regulatory challenges, some of which you'll hear about in the subsequent talks. And the important thing I want to say is, as a parent, you shouldn't feel that you want to forego standard of care therapies with the idea that, heck, I'm waiting for CRISPR, which is actually something or a sentiment that I've heard. Finally, I'm going to just put in a plug. I don't know Dr. Doudna. I get no royalties from a book or anything, but I will share this book, which I would recommend. It's reminiscent to me of the book, the famous book called The Double Helix, about the discovery of DNA, which itself is a flawed book. You may know it doesn't recognize the role, for example, of Rosalind Franklin in finding DNA. But this is a really engaging and informative and very thoughtful discussion, both about the science at a very lay level description of the science, but also discussion of the ethics and challenges. So this is the one that I send my family to when they ask me about CRISPR as well. So if you're interested in reading about science, I would suggest that this would be one reasonable place to start. So with that, we'll get started on our talk, and our first speaker will be Dr. Melissa Spencer from the University of California, Los Angeles. You all know Melissa, I'm sure, who's just a thought leader and a great scientist in muscle disease, muscle biology and disease.
Thank you. No one's ever called me a thought leader before. Okay, so I'm here to talk to you about CRISPR. Let's see. So this is an incredibly exciting technology that came around about 5 years ago. We knew about CRISPR in bacteria, but the ability to use it as a therapy actually became a reality 5 years ago. And so one of the reasons you may not understand why this is so significant is that we've always had the ability to splice DNA. We can take it out, cut it, put it back in. But what CRISPR does is it gives us the ability to edit DNA in live cells. And you can understand why the potential to edit DNA in live cells is so attractive for those of us interested in finding a treatment for Duchenne. What CRISPR allows you to do, many different things, but two specific things are, it allows you to cut the DNA and repair it with a new piece, or it allows you to cut it and seal it up without repair. These are two general things that CRISPR can do. So what I'm going to tell you now is how we can use CRISPR to develop a treatment for Duchenne. So first, let me explain to you how CRISPR works. It's a two-component system. So what I'm representing for you here, the blue squiggly lines, those are DNA. And CRISPR is a two-component system, and we're representing it with flags and scissors. So the flags are guide RNAs. Those are specific pieces of DNA that are targeted to anywhere in the genome we want. So we design our guides, flags, to pieces of the DNA, and then our flags guide the scissors to those regions. So this gives you tremendous potential to edit any part of the genome you want. So how do we use it as a therapy for Duchenne? If you think about the DMD gene as puzzle pieces fitting together, that top line, in a healthy person, their puzzle pieces fit together just fine. But in Duchenne, we have mutations that cause the puzzle pieces to no longer fit together. So you can see where that red circle is. The pieces are no longer fitting. And so we can target our guides, our flags, and our scissors to that region of the DNA, flank it, cut out the region that is not fitting together, and then the puzzle pieces fit together again. So it's similar to exon skipping, where you remove a region of the genome that is, or a region of the RNA in that case, that isn't in frame, and we're putting things back in frame, and you end up with a smaller, internally deleted but functional protein. So when you have mutations, such as the one shown there, where the puzzle pieces don't fit together, you have no protein production. And when you put the puzzle back together, you now have a smaller, internally deleted but functional protein. So we know that most DMD mutations lead to loss of protein production, and that there is an allelic disease called Becker muscular dystrophy that is due to in-frame mutations. And most of the genetic therapies that are being developed now are targeting this type of approach, are trying to take an out-of-frame mutation and make it an in-frame mutation, turn a Duchenne into a Becker. And one of the mildest Becker mutations that we know of is exon 45 to 55 in frame. And so, this region of the gene is also a hotspot of mutation. So I took this slide from the Sarepta webinar. Thank you, Sarepta. And you can see what it's doing is it's mapping all those mutations along the gene. And you can see there's a big peak where I've put those arrows. That's exons 45 to 55. And so that encompasses about 50% of patients. And so what we're doing at UCLA is we're trying to target that region using CRISPR. We want to mimic that very mild Becker muscular dystrophy phenotype. We know based on human patients who have Becker muscular dystrophy, this mutation is somewhat asymptomatic until patients are in their 60s. So there are patients diagnosed in their 60s, not every patient with this mutation, but we know it's one of the mildest mutations you can have. So this is our platform. So what I'm representing for you here with those blue exons is the region of the DMD gene we're targeting. And you can see those lightning bolts are representing our guide RNAs, or our flags. So the flags go to those regions that flank exons 45 to 55, and they bring the scissors to that region, and they cut, and then they bring exons 44 in frame or connect it to exon 46. And one of the nice things about our platform is that when you have cutting by Cas9, you get what we call indels, insertions and deletions. So you get bits of DNA taken out or bits of DNA put in. With our platform, it doesn't matter because we're in an intron, we're in a non-coding region. So that's one of the benefits. So the other two benefits, I've somewhat touched on them, are that we target about 50% of patients, and it makes a very, very functional dystrophin. So the phenotype is not just Becker, but mild Becker. So I'm showing you now, we generated a humanized mouse, so it has the human gene that's mutated on the dystrophic background, and you can see when we put our platform into this mouse using AAV in collaboration with Jeff Chamberlain, who's a speaker in our session, that we can restore dystrophin protein. So the red is dystrophin. You've seen this many, many times, I think, in this meeting. What I'm showing you here, that was skeletal muscle. This is the heart, and we can detect the protein. Whoops, I don't know. Is there a pointer on this? Guess not. You can see the Western blot, that we can detect the protein. It's smaller, but it's very, very stable. And it's smaller because we've taken out a really large piece. And so that was one of our questions. Can we even make this deletion? Because it's very huge. It's the largest deletion that anyone's made in the DMD gene. And we actually made our guides very far into the intron so we can encompass as many patients as possible. So you may be wondering, what is the difference between microdystrophin and CRISPR? Especially because they both could use AAV. So I thought I would just make a slide to give you a comparison. So when you're doing gene therapy using microdystrophin, you're adding in DNA. You're adding in DNA using a vehicle, which is AAV. But with CRISPR, what you're doing is you're actually modifying the DNA in the genome, and it's in its home, essentially. The gene is under the control of its normal promoter and regulatory elements, but you're doing a permanent change. And so because we're doing a permanent change, we have to be very, very careful about moving forward with this therapy. And we do have some concerns about using AAV to deliver CRISPR, and I've listed some of them for you here. One is that it has a very small capacity, as you've all heard. That's why dystrophin gene therapy is microdystrophin. And so what that means is to deliver CRISPR with AAV, you have to use 2 different viruses. And that's going to decrease your dosing. That's going to decrease the amount of virus you can put in because the virus is toxic. So you can only go up to a certain level. So by having to use 2 different viruses, you're effectively halving your dosage. and Jeff can correct me if I'm wrong on that. There's also an immune response to the virus, and so you can't re-deliver it. And so patients who might have the choice of doing microdystrophin or AAV CRISPR will have to make a choice, at least right now. We're hoping that we can devise ways to be able to re-deliver CRISPR or re-deliver microdystrophin, but right now we're limited to a single delivery. And so this is something to seriously consider because your therapy has to be quite efficient. Also, I am concerned, many of us are in the field, that this Cas9 is derived from bacteria. And we know that the episome from the AAV will stay around for about 10 years. And so we don't know what kind of an immune response might become elicited to this Cas9 bacterial protein in the muscle. So that's a serious consideration. And finally, there are reports that when the CRISPR-Cas9 cuts, the AAV will integrate right into that site. There are several reports of this. It may not be an issue, but it is something that changes the amount of control you have on the gene editing. So it changes the precision of the editing. So one of the things we've been doing is developing non-viral methods for gene delivery of CRISPR. And we are developing nanoparticles that could be delivered systemically. So I'm almost done. There are multiple forms of nanoparticles. They're made up of different materials. There are organic and inorganic. And the nice thing about them is they can be engineered and they can be repeatedly delivered. And there are now more than 50 approved nanoparticles, or nanomedicines we call them, mainly for cancer. And there are about 75 in clinical trials. So we're engineering these nanoparticles to optimize CRISPR delivery in vivo. The other advantage of them is they can be repeatedly delivered. So even if your therapy isn't super efficient, you can re-administer. You're not limited by that single delivery. And you can target them to things like stem cells. So we're doing all of these things at UCLA. And I've listed some things that need to be considered. So you can engineer them for blood stability. That's what colloidal stability. You can engineer them to cross the endothelial barrier. So they have to get out of the vessel into the muscle. And you can engineer them to improve cellular trafficking. So our platform that we're developing is able to target approximately 50% of DMD patients. We're now trying to directly deliver this CRISPR platform, and we're using nanoparticles that can be iteratively optimized and delivered systemically. So I just want to acknowledge our team, my colleagues on the top, including Jeff Chamberlain, who is going to be speaking, and the student Courtney Young, who just finished her PhD, Michael Imami, and Carrie Ann Stan, who many of you know, and our funding. So that's it.
Thank you, Melissa. As with other ones, we're going to save the questions for afterwards, after all three speakers are there or finished. Our next speaker is Dr. Dongchen Duan from the University of Missouri, who has devoted his career to muscular dystrophy and Duchenne muscular dystrophy.
I want to first thank the PPMD to give me this opportunity to present some of our results, and also appreciate the support from patients and family and PPMD and Pat and her organization in supporting us, although it's not directly to this research, but they have supported a lot in other work we're doing. So Melissa introduced some of the data showing that she can achieve the CRISPR therapy in cells, in human patient cells. So hopefully here I'm going to show some stuff that we did in MDX mice. I have a disclosure, and with Solid Biosciences. And this is not related to the CRISPR therapy. So this is how I view the dystrophin. If you consider dystrophin as a vehicle to use to bring you to the work or to any places, the normal lens dystrophin would be, the full lens would be like very fancy sport car, and this is what happens in patients. Your car is broken. And there's bigger patients. They have a car, it's useful but not terrific. And we all heard about that exon skipping and read-through drugs. Those like that you can rent a car because you have to pay the rent every day. If you don't take the drug, then you're in trouble. And we also heard about this microgene and metagene. They still work, but they're not as fancy as a car. So the CRISPR therapy is like trying to get you back to a BMD, but it's kind of like if you do it in your DNA, it's permanent. So it's kind of you rent that car. So Melissa has mentioned about that, and Kevin also mentioned about that. So in the CRISPR therapy, you basically have two components. One is your scissors, we call it Cas9. One is your guide. Your guide would bring your scissors to the location of the mutation. So we worked with MDX mice. Those mice has the mutation in the exon 23, so their messenger RNA are not stable, so they do not produce dystrophin protein. So what we did in collaboration with Charlie Gusbach's lab at the Duke University, so we used the guide trying to remove those mutated exon 23, and this is after removing this 23, you get exon 22 to 24, and you'll be able to produce a stable messenger RNA and dystrophin protein. It's slightly smaller. So first we looked at if we use AAV to deliver this component, can we get those to the place we wanted? So this is an immunostaining showing for the Cas9, which stands for with red color. And you can see that the Cas9 can really indeed get into the blue stand, those the myonuclear in the muscle cells, but apparently they're not a lot. So then we look at the muscle that's being edited, and if the exon 23 is removed, then we will see smaller bands over there. And we took the gene that is being treated and sequence it, we can see indeed that the intron 22 directly linked to intron 23. In between, the exon 23 is removed. So this is a Western blot showing that how much dystrophin you can get from delivering AAV CRISPR to the MDX mice. So on average, we get only about 7% of the wild type level. It's not super high. And this is looking at the immunostaining for dystrophin, and we can see fairly good expression in almost 80% myofibers. They tend to become dystrophin positive. And also, this editing restored NOS activity in MDX muscle. So then we look at the histology, and the top panel showing the HE staining. You can see that treated look much better, and the bottom two rows shows the staining for the markers of the inflammation for two cells, two different kinds of cells. One is macrophage, the other is neutrophil. You can see that after CRISPR therapy, those inflammatory cells are massively reduced in treated muscle. So then we measured muscle force. Comparing with the untreated the muscle, we see an increase in the muscle force, and also we see a protection against eccentric contraction induced force drop. So that what I just described to you is by directly introduce the CRISPR components using AAV to the muscle, to a single muscle. but apparently we need to treat all muscles, as Kevin has mentioned. So we tried to inject those CRISPR components in the AAV vector through the tail vein in the MDX mice. You can see that we see fairly good dystrophin restoration in both heart and the different skeletal muscle. So besides working in the mice, there's another important issue in the field is that we always worry about what if your Cas9, your scissors were cut randomly. You know, in other places, let's say it may cut your important genes that's essential for your life. So can we improve the strategy that currently being used to design this guide to make it more specific? So the current approach is to design those guide based on the base pairing of the nucleotide between the guide sequence and the region you want to cut. And so we took a different approach and did our study based on the free energy exchange. And without getting into details, I just want to show that we look at many different programs on there. And the bottom line shows that how successful your guide can predict off target. And this is what we got. So we're seeing improvement in this. So there are many ways. We're still at the early stage of CRISPR therapy, so there are many ways we can improve it. And so I'll stop here. And the funding was from NIMS and the DoD and Hope for Javier. And I also want to thank my collaborator, Charlie Gosbach, and the Xu Jiecheng group at my institute. Thank you.
Thank you, Dongshen. Our next speaker is Dr. Jeff Chamberlain, who you've heard mentioned before. I just want to point out as well, for those of you who were here for the gene therapy discussion yesterday, Jeff's name came up. Was in fact, he was responsible for the original design of the microdystrophin gene that's being used in the nationwide trial. So he was mentioned by Dr. Mendel yesterday afternoon. His career in Duchenne research and dystrophin research also goes back decades. So thanks for joining us today.
Thank you, Kevin. It's a pleasure to be here today. So what I thought I would talk about today is mainly to touch upon, as we've heard a little bit already, on some of the advantages and disadvantages of CRISPR-Cas9 editing. And the approach that we've taken in our laboratory is to try to identify some of the potential problems and begin addressing those problems. So I'm going to kind of give a general overview. I'm only going to show a little bit of data, but hopefully some of the points will be clear. One of the things in particular, maybe we should go back, is that most of the methods used to deliver the CRISPR-Cas9 machinery through the body right now are not unique to muscle, and the components go everywhere. And a problem with that is you end up getting gene editing in every cell of the body. And there, as Melissa pointed out, that can lead to issues with immune responses, where the body's immune system kicks in and tries to reject the machinery. And also there was a recent report suggesting that gene editing in dividing cells could potentially trigger cancerous events. And so we've been trying to focus on ways to limit the gene editing efficiency to muscle cells, which are less immunogenic than other cell types and also are incapable of forming cancer. So in general, my lab for a long time has been interested in gene therapy for Duchenne, with the goals of either finding a way to replace the dystrophin gene or more recently to try to repair the dystrophin gene. And of course, as you heard yesterday, gene replacement is usually these days is taking the form of AAV microdystrophin delivery. The gene editing is with the CRISPR-Cas9 system. And one of the limitations of CRISPR-Cas9 is that unlike a gene replacement where you have this one-size-fits-all shuttle that you can deliver, you really need to tailor the CRISPR-Cas9 system for every unique mutation that might be found in a patient. The goal, though, is to try to find a way to lead to more functional dystrophins. And depending on the mutation, very simple editing could lead to production of a dystrophin that's highly functional, but we should keep in mind that some patient mutations are not necessarily going to be editable or amenable to editing and may not be good candidates for that. So another remaining issue that Melissa also talked about is how are you really going to deliver this effectively? There are some limitations to using the AAV vectors that have worked out well for gene replacement. So, in keeping in mind the timing of some of these events, that as we've heard, gene replacement with the microdystrophins has already entered clinical trials. It's a little unclear how long it's going to take the gene editing to get into the clinic. I know there's some labs that may be fairly close to that, but I think we need to be aware of the limitations and not overly rush the technology. So, one question I get a lot is how does gene editing compare to microdystrophin replacement? And what are the pros and cons of the two? And should you wait for one or the other? And I think some of the critical features are that, of course, microdystrophin gene therapy is already in the clinic. 3 different groups have started clinical trials. The advantages of that are that you can achieve body-wide delivery with these AAV vectors. One treatment we hope is going to last for many years. It's highly protective against muscle wasting. And again, a single vector should work for all patients. However, there are some limitations. Microdystrophins are really small. It's only about 1/3 of the size of the normal protein, so it's not fully functional. The size constraints of these AAV vectors really limits how much you can pack into the vectors and how much you can deliver. And what I've tried to illustrate here on the lower part of this slide are the 3 different constructs that have been developed for the 3 different human clinical trials that are currently ongoing. And what you can hopefully tell from all those, is there a pointer on here? Oh, okay. All right. Every time I try to point at something, it changes the slides. But anyway, the constructs have a few common things and a few differences. They all have the beginning of the protein in common. They all have a region that we call the dystroglycan binding domain that is absolutely required for the function of dystrophin. But other than that, there's a lot of differences and variability between these different constructs. But the important thing to keep in mind is that you've noticed on the bottom part of those cartoons there's a lot of gaps. So there's a lot of regions of the normal dystrophin protein that are not present in these different microdystrophins. And the theoretical advantage of CRISPR-Cas9 is that you should be able to produce a much larger dystrophin, and that, in theory, will be a more functional dystrophin. But again, it's going to depend a little bit on the mutation that's in a patient. So a patient that has a deletion of the entire dystrophin gene is simply not going to be able to have that gene edited because the gene is not present anymore. But patients that have very small mutations should be, those mutations can be edited in theory and should produce essentially normal dystrophins. It should be highly functional. So what are some of the issues? Again, I think it's important to keep in mind there's really 2 types of editing, if you will, that CRISPR-Cas9 can induce. The most common one that I think most labs have been working on is really just editing, where you're trying to deliver the CRISPR-Cas9 machinery into muscle cells in order to modify a pre-existing mutation that's in a patient. So you're kind of tweaking the gene a little bit in order to take it from a state where it's not able to make this dystrophin protein to where it can make a partially functional dystrophin protein. Again, small mutations, point mutations, single exon deletions, things like that are going to be editable to the point where they will now produce essentially normal dystrophins and be highly functional. But again, other mutations are not going to be quite so editable because of either a large deletion or there are problems with critical portions of the gene. Now, the ultimate goal in many ways of the CRISPR-Cas9 system would be to actually repair the gene and take a mutant gene and turn it back into a normal gene. That is actually quite difficult, and I think a lot of the articles you see in the lay press tend to treat CRISPR-Cas9 editing as a repair mechanism of a way to fix a gene, and that usually is not the way that people apply this, and it's much more difficult to do. And actually, some of the work that Melissa was alluding to earlier is really, I think, the best example of work that's out there to try to actually repair the gene rather than to edit it and make it work a little bit better. So the focus that we're taking has largely centered around the idea of restricting the CRISPR-Cas9 machinery to muscle cells. And again, the reason for that is that we don't want to see gene editing in non-muscle cells because you don't need dystrophin in non-muscle cells. And also, there's a lot of side effects that can happen with gene editing with CRISPR-Cas9. And if that happens in blood cells or in liver cells, that has the potential to lead to uncontrolled cell division and cancer. So by restricting these components in a muscle manner, you can avoid a lot of those issues. The other thing that's going to be important is to try to adapt this technology for multiple types of mutations. As most of you are probably aware, Duchenne and Becker muscular dystrophy are new mutation syndromes. Every patient essentially has a unique mutation, and it's going to be necessary to design CRISPR-Cas9 delivery vehicles that will be suitable for every different type of mutation. So the way that we've tried to restrict CRISPR-Cas9 to muscle is to use on-off switches or so-called promoters that only work in muscle, that they don't lead to protein production in non-muscle cells. And an example of that is shown here on the top of this slide with an AAV, typical AAV vector that has small components in orange from the AAV genome itself, an on-off switch, and the one that we typically use is from the MCK gene, or the muscle creatine kinase gene, and that can control the production of either a protein like microdystrophin or an editing tool such as Cas9. And the advantage of this MCK regulatory element is it doesn't work anywhere except for muscle cells. It's inactive in immune cells, it's inactive in dividing cells, and in cancer cells. And Steve Hauschka, a colleague of mine at the University of Washington, has spent his entire career really trying to understand how this MCK gene works and develop very powerful gene regulatory elements from that gene. And in fact, all three of the microdystrophin trials that are currently ongoing use components of the MCK system that Hauschka has developed. So we've been trying to adapt this to the CRISPR-Cas9 system. And just here's an example. One early question we had was, if we limited the CRISPR-Cas9 machinery only to muscle, would it still work? We were worried that it would be too weak and it wouldn't lead to dystrophin production. In fact, if we inject these vectors right into muscle, it works pretty well. We can induce exon skipping. We can, in fact, induce gene repair and completely repair the gene in mouse and produce a fair amount of dystrophin, up to 25% of normal. But this is kind of an artificial approach because we're putting these vectors directly into a single muscle, and that's not going to be a therapeutic approach. As Dongsheng Duan alluded to earlier, you really need a systemic delivery, so we've begun looking at delivering these same vectors body-wide by infusing them into the bloodstream, similar to the microdystrophin trials, and this is an example of what we see here. We see actually a lot of variation from one muscle type to another. We don't really understand why that is. The best gene editing that we see is in the heart, so that's a very good target. Some of the limb muscles don't get dystrophin production as much, but with the systemic approach, we're typically achieving less than about 10% of normal levels of dystrophin. Another thing we've done is we've tried to start scaling this up to larger animal models. So we've been doing some gene editing in the canine model, the dog model for Duchenne muscular dystrophy. And part of this was to try to ask, will this work in species other than mice? Most of the gene editing work that's been published has been done in mice or small animal models. When we go into the dog, we do see similar levels of gene editing as we saw in the mouse model. But if we do a side-by-side comparison between gene editing with CRISPR-Cas9 and delivery of microdystrophin, the microdystrophin is still working much, much better. So just to summarize, the muscle-specific gene editing approach that we've taken limits gene editing to muscle tissues only, non-dividing muscle cells. You can produce very large dystrophins, but again, it depends on the mutation, and it seems to work moderately well on both mice and large animals. But there are a number of things that still need to be improved with this system, in my opinion, to bring it into the realm of clinical trials. One of which is if you really want a permanent cure of the disease, you have to go beyond treating muscle, and you really need to find a way to also treat the muscle stem cells, because muscle stem cells provide a lifelong source of new muscle as muscle slowly turns over throughout life. So far, people haven't found good ways to get gene editing in muscle stem cells. There's still a number of safety concerns, potential links between CRISPR-Cas9 delivery and immune responses, and possibly in the induction of cancer if you get editing in non-muscle cells. And this comes from unwanted editing of other genes. You get a lot of non-specific effects. It's not quite as specific as we'd like it to be. And as Melissa was talking about, AAV, I don't think is going to turn out to be a very useful vehicle for delivering genes. It's great right now. It's the only way we know that we can deliver genes body-wide, but it lasts for a really long time. We're hoping that AAV gene therapies will last 10, 15 years, maybe longer. You don't really want this CRISPR-Cas9 machinery sticking around in the body for that long. So we need to find other ways to deliver it that are going to limit it. And ultimately, it'd be nice not to just be tweaking the gene, but to completely repair it. And that's proving very difficult to do. So thanks.
So we will, I want to thank our speakers. Everybody intentionally kept it short so we'd have plenty of time for questions. So if you have questions, please come up to the 2 microphones on either side. And I'll vamp for a minute here till we get people up here. And we'll do what I learned from Dr. Timothy Kripe yesterday. We'll do 2 in a row per side if we have a line at least, so our cameraman doesn't have to run around. So why don't we start over here? You have a question for one of us? Yep.
Hi. My question is, I know there's several research already doing on CRISPR-Cas9. I know Dr. Olson's already doing it, and I think he already have done it with dogs or something like that. So I want to know what would be the difference between all you guys doing this and him, or if you guys are working together, or how that works.
Well, there's a number of labs currently applying CRISPR-Cas9 technologies to Duchenne muscular dystrophy. Eric Olson is probably the leader in the field, as you point out. Everyone is taking a slightly different approach. There is a lot of crosstalk. We do interact. For example, the vectors we've developed that restrict the CRISPR-Cas9 machinery to muscle, we have sent to Eric Olson's lab, and that's now the system he's using as well. You know, it's important, I think, though, that we try as many different variations as possible. So while it's great that everyone is talking, they're still trying slightly unique things in the hope that eventually this will start working very well. One of the things that I think has proven to be quite successful with Eric Olson's approach is he's limiting his approach to really single mutations, where he's just trying to target one single part of the genome, and that turns out to be a lot more efficient than some of the other more complex strategies that you might need to actually repair a gene or to modify very large deletions or complex mutations. So he's kind of taking the simplest approach, which makes a lot of sense if you really want to advance the technology, and it's coming along well. But I would say the approach he's taking is limited to a very specific subset of the mutations in this disease.
I had the opportunity to talk to him last year in Austin, and I talked to him about that because our boys, the mutation is 10 and 11, and the more common is on the 50s. So when I talked to him, we talked about that, and he said that they wanted to focus on this, kind of like make it perfect, so then they can go and focus on the rest. But he said once they do that, they'll be able to know if it works for the rest.
Yeah, I think that's true. It's a complex system. It's not quite there yet. There's still a lot of work to do. So you know, anytime you're dealing with a situation like that, it makes a lot of sense to sort of narrow down the number of variations and questions that you need to address and see if you can get something working really well in a limited set of conditions, and then see if you can expand it beyond that. And that relates a little bit to what I was alluding to earlier, in that you know, there are thousands of different mutations that have been found in different patients with Duchenne muscular dystrophy, and the CRISPR-Cas9 is not a one-size-fits-all system. It's going to have to be individually tailored for many, many different mutations, and it's probably beyond the capability of any lab to go after all of them at once.
Okay. Thank you.
We'll do another one over here. The question here.
So thank you. I'd like to comment on a point that you made early in the discussion, Dr. Flanagan, relating to putting life on hold while we're anticipating some of these therapies. And I recall attending a conference about 15 years ago with Dr. Luke Kunkel was explaining about a patient who had leukemia, and there was a bone marrow transplant. And I think Dr. Chamberlain probably is more aware of this this research or this discovery. And the individual apparently had expression of dystrophin because of the closeness of the bone marrow transplant. I believe it was from a parent. And how during that particular presentation, there was a parent who stood up and asked why they couldn't go to their physician and have that same procedure done. And there was a rather lengthy explanation about some of the caveats and some of the dangers with bone marrow transplantation. And so it's 15 years later, this particular treatment never came to fruition, but I think that it does lend itself to a cautionary tale about continuing to focus on standard of care while we're anticipating some of these promising treatment opportunities. And I just wondered if there was a comment from the panel about that concept.
Well, I'm old enough to be able to say that I was here from the beginning, and that is the case with every new idea that comes on the table, that it's always promising, it's always more promising in the mouse, and we always encounter many obstacles along the way. And so Jeff Chamberlain and Dongsheng, but I remember Jeff starting microdystrophin 2 decades ago, and now we have it. And it looked like a very simple thing. You just put the gene back. Why is that so hard? But here we are, and it did happen, but it took development. It is not a one step, and it takes a lot of people and a lot of funding. And I do want to express my gratitude to PPMD, to Pat, because I also remember when there was nothing. There was no parent group, there was very little funding for Duchenne, and all of your efforts do matter. And the work of Jeff Chamberlain here, and Jerry Mendel, and everyone who worked on these therapies, it all does add up, but it is not overnight. And so what we have right now are some exciting therapies. And I think waiting for CRISPR is not a good idea, that it is not tomorrow or even in a couple years. But we have amazing options right now, and we're altering the course of this disease, and that's terrific.
If I could just add to that, I wasn't kidding that I've heard in clinic that sentiment. People say, well, we're wary of steroids, we're aware of the side effect profile. I'm going to wait for CRISPR, and I think this point and your point is correct. These are being studied by a lot of laboratories. The combined effort of these laboratories are going to put us in a place we can't even predict, really, 5 years from now where we'll be. But clearly, the standards of care aren't altered. That's the keystone to care for patients that we have. Being on a reasonable steroid regimen, doing all the other issues related to cardiac care, pulmonary care, bone care. That's the real touchstone of care for Duchenne. Keeping your boys as healthy as they can be until these reach fruition, until they're safe to give. That's important. And there are particular safety issues. They've been touched on here a little bit. The safety issues are distinct when we're intentionally going into altered DNA. Dr. Dwan mentioned that if we nick or damage a critical gene, say a tumor suppressor gene, then we wind up with cancer. I mean, these are very significant risks. Many labs are working to mitigate them, among other reasons, by using expression systems that don't continually express this nuclease activity for 10 years. That's probably not the best way to do it. So we'll reach that point, but standards of care haven't changed and won't change while we work this out.
The only thing I would like to add is that regularly I see people make recommendations that people disrupt standard of care with the advice to others that they consider enrolling their child in clinical trial. And that to me is poor advice, because a clinical trial is not treatment. It's a study. It's to determine whether or not something is safe and efficacious. And so I would urge people, and that the point I'm trying to make is that adhering to standard of care while we anticipate many of these treatments. And I think through every one of the discussions I've heard over this week has been very, very optimistic, it's very promising, but I don't think I would put my, my money on any one, because I think collaboratively or collectively, that's the approach we're going to need to take. Thank you.
Thank you for those comments. We'll move over here. Sorry, we'll give you a second.
Thank you, everyone. You compared gene editing or CRISPR-Cas9 with microdystrophin, but how do you compare, or in terms of therapy, with exon skipping? I mean, with the length of the dystrophin, and is exon skipping a better therapy compared to gene therapy, or any thoughts on editing versus?
Any thoughts? You'd like to, Duchenne, do you want to?
Sure. I mean, Jeff has already showed the data comparing the microdystrophin and the gene editing in the dogs, and there's apparently different. At the beginning of my talk, I showed those fancy cars and different things. So exon skipping and the gene editing, there's some similarity. So they both aim to remove the mutation remove the mutation from the. So in the gene editing, you do this in the DNA. So what the protein medicine initially have a DNA, then this DNA make messenger RNA, then this messenger RNA translate into a protein. So in the exon skipping approach, so you're targeting the messenger RNA, not the gene itself. So the gene is still generating mutated messenger RNA. So that's kind of why for the exon skipping you need the repeated, administration of the oligos, Anderson's oligos drugs. And so in terms of the microgene and the gene editing, and Jeff has mentioned that the microdystrophin has only a size of about 1/3 of size of the full-length protein. And as Jeff showed in his cartoon, that comparing the full-length dystrophin versus microdystrophin, you're seeing that the microdystrophin are missing a lot of components in there. So we don't really know that, the nature make this full-length gene for a reason, right? So there may be pieces that we'll never be able to put in there in the context of a microdystrophin. So I would say that microdystrophin would, improve. It's going to be better than no dystrophin, but it's not going to be a cure.
Correct. Actually, my question was really in terms of a length of the dystrophin, which is going to get reduced in terms of microdystrophin versus exon skipping therapy. I mean, I'm assuming since it's only going to skip one exon or two exons, it's going to be length is going to be bigger and maybe more beneficial. Or am I wrong?
Maybe I can address that. So I think. To take for comparison, the first thing we should say is with exon skipping, let's take exon 51 skipping, for example, as a therapy. All of the outcome genes, the proteins that we get out of that are not all equivalent, right? Because if you have a boy with 45 to 50 deletion, they can be skipped by exon 51. You get a 45 to 51 skipped gene. And a boy who's missing exon 50 alone, you can skip that with exon 51 as well, and you get a 50 to 51 gene. So those proteins that you make from those two are not exactly equivalent, right? So they're slightly different sizes. We can anticipate they might have slightly different function. We can anticipate that they are both better than no dystrophin at all, but they might have different outcomes. And what we actually do is we look at patients with Becker muscular dystrophy to say, well, what's the best we can expect? If we see someone with Becker walking around with 50-52 or someone walking around with Becker with 45-51, that's probably the best we can get with treatment with those. Now, Dr. Spencer's approach, she's describing that approach where he says 45 to 55, even though it's a bigger deletion and a smaller dystrophin protein than exon skipping. We know from patients, as she described, who into their 60s with that, walking into their 60s with that mutation. So it's a very, although the size is different It's a highly functional protein. There's critical domains there that are functional. Same with microdystrophin. The microdystrophin construct that Dr. Chamberlain first designed and is in one of the trials now, it's smaller yet, but it has critical, essential domains to it. So my opinion, for whatever it's worth, is which of these is going to be better? We won't know until we get them into human trials and see in the human setting how they actually compare. So that's a great question, but I don't think, personally, I don't think it's one we can answer yet. But there's a rational approach to 45-55 skipping. And another question about specific mutations in Dr. Olson's approach. Each of those are going to be somewhat different as well. So it's not quite an apples and oranges, or it's kind of an apples and oranges comparison, if that's helpful.
Thank you.
You're welcome.
Thanks. I got it.
You got it?
Hi, my name is Yuva. I'm from Philly, and I'm going into 11th grade. And before I ask my question, I just wanted to do a quick little aside. I thought, Mr. Doug, your analogy of all the different, with the cars was really helpful, and I thought it was really cool.
Thank you.
And now my question. So I know with postnatal AAV mediated, you're using AAV, the virus, to deliver CRISPR, but after this virus goes into the body, from what I understand, neutralizing antibodies are made. So this is like a one-time thing. Like the AAV, once it goes into the body, it cannot be used again with the antibodies. But then learning about the nanotechnologies, the gold nanotechnologies, as a mechanism to deliver CRISPR, I just wanted to know, could antibodies be created in this situation to these technologies?
Before I let Dr. Spencer answer that, I'm just going to say, you're going into the 11th grade. Would you come work in our lab next summer? That's,
So we can design these nanoparticles many different ways, including adding proteins to the outside to target them. We can coat them with polymers or lipids. So it is possible, but the nice thing about them is you engineer them. So if you see an antibody response, you can alter them to reduce the inflammation. So it's actually not going to trigger. It's not going to trigger the same kind of inflammation or rejection that a virus would. It's different, but it could be seen by the immune system and coded, for example, by antibodies and removed. So the answer is they could, but we can also engineer them if that's the case, to alter that.
So by making small changes to the technology, you can avoid responses to antibodies that have already been created?
Yes, beautifully stated. Okay. Much more simply than I did. Congratulations.
Thank you, guys.
Are there any other questions? Dr. Wong has a question.
Thanks, Jeff, Melissa, and Duchenne for a very informative talk. So I've got a question. So if we were worried about altering permanent changes in DNA, what about CRISPR-Cas9 on RNA?
Jeff, you want to?
It is possible to do, as you probably know. I mean, really just in the last year, a couple of laboratories have published modified enzymes that will go after the RNA and edit it instead of the DNA. You know, there's some advantages and disadvantages to that approach. As Dr. Dewan was talking about earlier, the issue with editing RNA is that the RNA is continuously being produced, and you're going to have to continuously edit it throughout the lifespan of a patient, similar to the exon skipping approaches. Whereas with genomic DNA, you're actually trying to go in and more or less permanently repair the gene itself, so that you can kind of have a hit and run mechanism. You go in, you do your change, and then everything should be nice afterwards for years and years, hopefully. So there are potentially some safety considerations that RNA editing could be a little safer. You're not likely to induce a permanent bad change if something goes wrong. But it will require lifelong production of the CRISPR-Cas9 machinery, which can have some significant immunological problems.
So we have 4 minutes left, so we'll just do 3 more questions, 1 or 2 over here and 1 over there, if that's right.
Hi, this may be a question for Dr. Flanagan, or actually I'm asking you for a comment in a sense. We had someone ask you to compare exon skipping, microdystrophin, and gene editing, and I just wanted someone to comment on to what extent we think exon skipping impacts cardiac muscle, and I just wanted that to someone to clarify to what extent we are expecting some benefit in cardiac muscle.
So I think the standard technologies, the PMO technologies, have not been as conceivably are not the final word of this for cardiac muscle. I'm not sure if you saw any of the data, the PPMO data that was presented, but the peptide targeting for exon skipping, at least that Sarepta has presented, I know that data best. It certainly looks like it has much better cell penetration in the heart and better expression in the heart. And there are other exon skipping approaches that are virally based exon skipping approaches, for example, that are being developed that show great expression in the heart as well. So we'll get there, I hope.
Thank you. So this question, so I understand, we are a few years away from the clinical trials, but I'm wondering if you're able to comment on the possible design approaches for these trials. Like, do you see them following a pattern that we see for exon skipping, where certain mutations or certain hotspots are targeted first, and then certain mutations, at least we don't see them being targeted today? Or do you see them, is it possible to do like a platform approach where all mutations can be targeted? I believe some of these points were addressed or touched upon. I'm wondering if you can elaborate a little bit more on that. Yeah.
We anticipate that each platform will need to be approved by the FDA in a similar manner as the exon skipping. It's one of the reasons why we chose a single pair of guides to target the largest number of mutations with our platform. But we hope that eventually there will be a path like with vaccinations, for example. Mm-hmm. So once you have some approvals under your belt, that the FDA will start approving them more quickly once these technologies become more commonplace. That's my expectation, but maybe someone else could comment further.
Well, yeah, I mean, I would agree with that, and it gets back to the point that we're making earlier, in that genome editing is more in the realm of personalized medicine, in that with everybody having a different mutation, there's not going to be a one-size-fits-all treatment, and so those are going to have to be individually tested and approved by the FDA to a certain extent. However, as Dr. Spencer was pointing out, the system she's working on potentially could be a single system that would be applicable to up to half of all patients, which would be fantastic. So it may end up in the realm of a lot fewer drugs, if you will, than we may be thinking about now. It's going to depend a little bit on further development and how these things fall into place. The way things currently stand, the most efficient scenarios are ones that are targeting individual mutations. And if it stays that way, it's going to take an awful lot of individual platforms to treat everybody. But I think we're seeing the writing on the wall that that's going to change, and that eventually we'll get to the point where there'll be a handful of platforms and not dozens.
Okay, so thank you. I think we'll take there's 2 standing here, so we'll take the last 2 questions. I'll beg the indulgence of the next session, and we'll just take the last 2 questions here.
So my question is for duplications with CRISPR. Is it possible to only cut the duplicated region while leaving the original exons there? Or if you cut it with CRISPR, the original and the duplicated exons, they all get cut?
Can I take that? So it's actually quite promising to consider duplication mutations depending on how big the duplicated region is, depending on whether you can get unique targeting sequences or not, whether you're doing a single exon or the size of those duplications. But one very promising approach, it's actually been demonstrated in at least one cell culture model, is to take a single exon duplication and cut it out by use of CRISPR technology, leaving a wild type, completely normal dystrophin protein. But it's not a one-size-fits-all question. It really depends on if you have duplicated the regions that you actually want to target, there can be a real challenge to it. But it holds great promise for duplications.
Okay, thank you.
And our last question.
Hello, and thank you to everybody. I have a question about nanoparticles. And I would like to know more about the target strategy of nanoparticles, how it can be specific, and what is the capacity in terms of, does it should care a genome, or does it should care a protein itself, how it works?
So we're looking at different ways to deliver the CRISPR in terms of the platform itself. So we can use a plasmid, so DNA. We can use RNA, which is actually more attractive because it will go away quickly. It won't hang around. Or we can use something called an RNP, which is RNA plus protein. So you deliver your flags plus the scissors together. And for each of those platforms, it's going to require a different nanoparticle. So we are trying multiple approaches, and we're optimizing them. We have designed now at least 8 different formulations. And our goal is to administer them systemically, so through the bloodstream. And we want to target them to skeletal muscle, ultimately target them to the muscle stem cells. We aren't there yet, but that's our goal. So we're trying everything. I have a team on it.
Thank you. All right, well, thank you very much, and thanks very much to our panel members. Thank you.
It's always a little scary walking up to a podium when everybody's walking towards the door, but I'm getting used to that. So I did convince Kimberly to put me between Kevin Flanagan and Pat Furlong in hopes that somebody would listen to what I had to say. Anyhow, I'm John Killian. I'm the chairman of the board at Parent Project Muscular Dystrophy. For those of you who don't know me, my son Sam, who's tooling around out here, is 16. So we've been part of this organization for quite a while. But I just wanted to recognize a few people before Pat closes out the conference, and start out by saying this is and has been an incredible conference. This is my 14th conference, one of which I've watched on a monitor, so anybody watching at home, I've been there and done that, but all the rest I've been here in person. But it's our largest conference ever in almost every way. Over 600 people here, the most new families we've ever had, the most researchers, the most things to talk about that we've ever had. So a lot of excitement in the room. I'm obviously very excited about that as well. But this is the time to stay focused. This organization will, you've heard all these people up here talking about staying focused. We can't rest on the fact that we've had some success because there's still a long way to go. But I wanted to just recognize, first of all, the sponsors, some of the sponsors. We have a ton of sponsors. A lot of folks in the audience here are elite sponsors from PTC, Sarepta, and Santhera, who put a lot of effort and resources into bringing people here. So I hope that you'll take the opportunity to thank those people, not just drill them for answers out in the hallway like we've been known to do. But I also want to take the opportunity to thank the staff at PPMD. These people work tirelessly for our sons. They are as engaged as anybody in this community, and I hope that you'll take the opportunity to thank all the people that you see come up here, Pat and Kimberly, Annie, Abby, Ryan, but also the folks at the desk who are here every day. We don't pay them overtime, sorry guys, but they really are engaged in this and care about this community. So I hope you'll take the opportunity to thank them. Yeah. Ryan thinks that's all for him because he's sitting on stage. I also wanted to take just a minute and mention our board members. So, the people on the board, first of all, we do put a lot of effort into trying to help translate what the parents and all of us in this room want to see happen, and we should be the liaisons to the staff. So we're here for all of you guys, so always feel free, and some of you do, some of you don't, feel free to reach out because I think everybody on the board wants to hear from people in the community and wants to play that role. So just in case you don't know who the board members are, some of them are in this room. If you are, please stand or wave your hand. Ellen Wagner, Chris Curran, Chris Jones, Rasha Albaneri, Joanna Johnson, and Christine Piacentino, and then Dr. Jonathan Finder and Dr. Linda Kripe. They bring a little bit of brain power to the board. So really encourage you to reach out to those people and give them your ideas, your opinions, and spend some time with them. But I want to close this with one reminder. Over the years, I can't tell you how many people have told me we should change the name of this organization. Parent Project Muscular Dystrophy just doesn't sound that professional. You don't want to be just a bunch of parents. Well, we're not. We're not just a bunch of parents. We're involved. We know about our sons. I think if you talk to the researchers, to the clinicians, to anybody involved in this, they know that we're here for the long haul. We know what's going on with this disease, and I encourage you, don't tell me to change the name of the organization. Be the parents who get involved. Find a way to get involved. If you're a fundraiser, go fundraise. If you're not a fundraiser, go find a researcher that you can help support in another way. Call the office. Ask them if there's anything that you can do. It's called Parent Project Muscular Dystrophy for a reason, because you all are the most involved people here, and we've come a long way. We've got a ways to go, but I'm really excited about where we are. So thank you all very much for being here. I'm going to introduce Ryan, who's going to do his last polling. He's so excited about this polling. So anyhow, thank you all very much.
Okay, you're probably sick of me by now. So the next couple of questions are actually for members of industry that are hopefully still with us in the room or perhaps listening online to participate in some of this polling. You heard a lot about the great research that's happening and the clinical trials underway. There are so many underway that the institutions certainly are overrun with them, and we know that there's challenges associated to doing those trials. So the first question is for industry about conducting trials. We want to understand, in your opinion, the top 3 barriers to conducting those clinical trials, and I'm hoping we have some industry representation in the room And so far, it looks like we don't. Oh, is that the one person in the room? Okay, there's, okay. All right. We see there's numerous barriers to conducting these trials. Good, that means there's people in the room from industry. That makes me happy. I'm going to give it a minute. All right, this is a real widespread of these barriers to doing these trials, as we can see. As more companies are coming online, there's a need to really look at these barriers. And the patient recruitment, participant recruitment is high on that list, followed by study site activation, challenges in endpoint selection. Okay. Still going. Okay, I think we can move on to the next question. In this one, we talked to many of the companies before this to ask to represent one representative from each company answer this question. Are you currently conducting studies in non-ambulatory patients? And okay, I was going to say 100% can't be right. And that's not if you're planning on conducting these studies, it's if you're conducting the studies currently. And this is one of those questions that I think we're going to have to really send out to the full list of industry partners, because if they're not present right now, it's not giving us the most accurate count. But we see that in this poll anyway, 45% are conducting them. And I promise you, when we post the final results of these, we'll make sure that all of our industry partners answer this question so we get an accurate count. So the next question, do you currently have a public statement posted about access to unapproved therapies? So expanded access, compassionate use, or now right to try. Do you have this, and is it made public on your website? Still coming in. Okay. So for the folks who said not at this time, it's now been put in legislation that you need to make public your policy on access to unapproved therapy. So we're happy to chat about that. Okay. Still coming in. Okay. Oh, some people still thinking about it. All right, so let's move on to the next, should be a word cloud. Oh, this is another industry question. I'm sorry. For those studies that include non-ambulatory patients, does that include adults over the age of 18? Important question, because quite often the adult population, 18 and above, are not included in these studies. This is, again, one of those questions I think we need to go back and revisit with the full list of drug companies within the space, because if we have a limited sample here, it's not giving the most accurate picture. But it's good to see that for those companies represented right now, there are a percentage saying that there is 18 and above included in that inclusion criteria. Okay, we can move on to families from your perspective. This is for any family member, so it's your turn here. What are the two of the greatest needs in current clinical trial landscape from your perspective? So go ahead and choose two. We see that placebo item often chosen. Broader inclusion criteria. give it another 30 seconds. So you can make your decisions. Still coming in. Ten more seconds. We see that broader inclusion criteria is by far in the lead, followed by a similar theme we hear throughout the conference about limiting the use of the placebo in trials. All right, John, do we have the word cloud question as well, or is that saved for the end? Okay. Okay. Okay, this is a word cloud. We're asking you, based on your the care meetings that you participated in, give us one word about care that you will manage differently when you get home. This is kind of cool. We see physical therapy, nutrition, constipation. Y'all just wanted me to say constipation, didn't you? See physical therapy, nutrition. I like this word cloud thing. Why did we wait to the end to do the word cloud? This is really cool. Behavior and learning. People are still making choices. Pulmonary, I see pooping on there. PCA, dental, physical therapy, nutrition are really a consistent word used throughout, and constipation. And gratitude. You're going to work on gratitude. I love that. From Chris Jones' remarks earlier. It's really neat. Okay, with that, we are going to invite our final panel up, led by Pat Furlong, talking about family dynamics and Duchenne muscular dystrophy. Thank you very much.
Okay, so this is the very last panel of the day, and it's always kind of bittersweet, right? We've been together for more than 3 days for some of you. I am quite certain that a lot of you have bottom fatigue, which comes, right? So our last panel was really about stories, right? You heard from Chris Jones today, and he talked about his story, which I think is incredible, but all of our stories are incredible. So I'm going to just get some opinions. We're not going to take questions, we're just going to chat. So that's why I had everyone sit on one side, so that I only had to stare one way. The yoga didn't help my neck this morning. So we're just going to talk about our experience and their experience. So first of all, I'd like you all to introduce yourself. So I'm going to start with you, Braden, and you tell us why you're here and how you got here. Well, you don't have to tell us that you flew or drove, you know.
I'm Braden Hester, and my brother has muscular dystrophy, so that's why I'm here.
Good.
I'm Diane Fryer, and I had two grandsons with Duchenne.
I'm Mike Gaglione. I have a 22-year-old son, Michael, with muscular dystrophy.
I'm Colin Wrench. Can you guys hear me? Or do we need to lift the mic up? Good. And I'm 24, and I have Duchenne, and I'm on the Adult Advisory Committee.
I'm Gretchen Egner, and I have a 19-year-old son, Nick, who has muscular dystrophy.
Hi, I'm Dee Dee McGee Castle. I am the stepmom, or mama too, of Anthony Castle, who's 18 years old with Duchenne muscular dystrophy.
Good, thank you. So Mike, I'm going to start with you, and can you just tell me a little bit about the diagnosis and what the diagnosis meant to you?
So we were diagnosed when Michael was 5. Obviously, it was, devastating, at the time. There was a lot of, crying and everything like that. But, we have we decided from the very beginning, that, we were going to have a very positive outlook as far as how we, raise him and find the best care for him. And that's how we find PPMD. So, I was just talking to my wife about it. We said, we have a common enemy now, and we have common goals. So, that's kind of how we look at it. We try and be positive, with our son. We have him do things that maybe he wouldn't have done, at the ages that he's done things. So.
Good. Yeah, I saw you put him on the edge of the Grand Canyon.
Yeah.
We can talk about that later. Gretchen, can you tell me about the diagnosis and what it's meant to you?
I think number one, initially when he was diagnosed, it meant safety and everything that was things that I could take for granted. He was my second kid, and so I wasn't going to be as high-strung about it, and I was more laid back. And so then I had to regroup quite a bit. And then it just means that I need to be more concerned, or when he was younger, I needed to be more concerned about falling and making sure that in his everyday life things were safer, but then also trying, as Mike said, to give him the most normal life possible.
Good. And Colin, tell us what the diagnosis meant to you.
Oh, well, for me, I was diagnosed when I was 3, a little closer, yeah, I think that'll be good. Yeah, I was diagnosed when I was 3, so like, many kids with Duchenne, I probably didn't think too much about it, just I was a little bit slower, things like that, until I was, maybe 7 or 8, and then I think I got, finally heard the word. But, it's meant a lot of different things, a lot of, struggles, a lot of obstacles, but also a lot of interesting opportunities to see the world in a different way than I probably wouldn't have gotten to, if I hadn't had Duchenne. And I'm also a problem solver. I love to solve problems. I'm also very creative. So it's, I really appreciated the opportunity to kind of figure out some solutions to some issues. And that's really what you have to do. I mean, because you can sort of spend your time being bummed out, but, that wouldn't really be, and that's kind of who I am too. So yeah, it's meant really that there's more hurdles in my life, and I've seen life in a different way, and for my family too. I think we've all, the experience has taught us all a lot. So.
Thank you. So Braden, I'll pull the microphone over by you if you don't mind. So Braden, so you're this healthy brother, right? And tell me what the diagnosis did to you and how you responded.
Well, I was about 5 or 6, I think. Yeah, my mom's nodding her head. I didn't really, at first I didn't really understand, but I think it really sunk in when I was in elementary school with Micah, because I would see other kids like he would be walking and not running like the other kids, and so at first I was very protective. I still am, but more aggressive. If kids were mean to him, I would threaten to beat him up and stuff like that. And then I think once like middle school and high school hit, I was more like, I don't know, like relaxed about it, just because Micah has a lot of friends and like we have a lot of support. So it always meant like, yeah, it sucked that he had this disease, but it brought a lot of families together in like trying to find a cure. So I think that I kind of just grew up knowing that it was something that a lot of people were fighting to find a cure.
Did you gain or lose friends?
I think I gained friends just because a lot of my friends enjoy Micah over me. So, yeah.
So, Dee Dee, mama too, right?
Yeah.
So, can you just give us a little insight about a lot of marriages fall apart in Duchenne muscular dystrophy for lots of reasons. People grieve differently, they have different opinions, and marriages fall apart just anyway. So, you came on the scene as mama too. So how did you get into this family, and how did that affect you?
Well, I've been blessed to get into this family, and just us being open. When Chris and I got together, and I first met Jill, and I think it was about putting the boys first and making sure that they were okay. And then, and then working on us. I knew Anthony was different, but I wasn't sure exactly what it was. Chris is not the best at giving the best descriptions, but when I met Jill, I got it all. I mean, I got every bit of it. And but it was great. It was informative. I understood better the struggles, because I had a better idea about it.
Thank you. Yeah. So Diane, you're a grandma, and sometimes I think as a grandma, because you care so much for your kids, and then you have a grandchild who's ill, and as maybe some mother-in-laws or mothers have opinions that are different than their children, it's hard to get in there, right? It's hard to know when to zip your mouth or when to open it, and it feels sometimes double pain when you're watching. Tell me how you feel about that.
Well, I had to step back sometimes. I try really hard to wait until I'm asked for an opinion or a thought, or to enter into the conversation. If you ask my son, he's going to tell you that that's not true all the time, and it's not. But I'm not there. I don't live it 24/7, and so I don't understand all of the stresses and all of the needs that go on exactly. I'm there a lot, so I do understand a good part of it, but I just try to wait to be asked. And then I do give my opinion and my thoughts.
I'm a grandma too, I'd never give my opinion. So Mike, where do you find strength?
Where do I find strength? Well, that's an easy one. Michael, I look at him, and what he goes through every day, and I think that is, kind of cliche, because that, everybody could say that, but it's true. I'm there to help him, and I can, it's hard to try and put yourself in their place, but you really can't. So that's, if he can do what he's doing, then that's, I can do whatever I need to do. So it gets me through a lot, yeah.
Did you lose or gain friends during this?
We've always had a really great group of friends. So I would say I've gained a lot of friends, and a lot of them are here, But, and I know it's not true for everyone. I know people that have lost friends, and for me, it's kind of mind-boggling, but, we have a really good group of core friends that really support us a lot, so.
Colin? Where do you find strength?
Oh, well, I find strength, well, you know, just with the, talk about a couple things, but the first is the PAC. It's been great being on the Adult Advisory Committee. It's a great group of people, and, you know, it's really been great to kind of be trailblazers in a way, and the, you know, adults finally, living lives past their 20s and doing, you know, great things, and that's really been a great part of it. And our fearless leader, Annie, over there, she's been just fantastic, and we've had just lots of good. There's been some ups, some downs, some times when we were sad, but we're a good group. Beyond that, from my family, a great close-knit family. I just have one brother, and then my mom and dad, and we're just really close-knit. And you know, we do things together all the time, and growing up, you know, that was never an issue. You know, if Henry might do his stuff every once in a while, but as a family, you know, we'd go on vacations together, we'd, you know, go parasailing, Henry and I mean, we'd try to do stuff, you know, go skiing as a family, you know, I was in my sit ski, so, but beyond that, I draw a lot of inspiration from music and from my faith. Those are really important to me, so.
Thanks, Colin. So Gretchen, this is a struggle. Has it been a struggle for you, and what have you done?
Well, I keep my sense of humor, and I try to keep things in perspective. I have amazing friends that I've met through PPMD. I thought you were going to ask me the same draw strength question, and I was going to say, well, this place, because I remember when Nick was first eligible for the Adalurin trial, and only 13% of the boys have the premature step, and your first thing was, what about the rest of the boys? And to see how this has grown is really amazing. But I run, and I you know, have friends who aren't affected by Duchenne directly, and everybody has problems. And so being able to realize that even though we have a predicament that is quite stressful, we can be empathetic to others. I saw gratitude was pretty big up there, and I think living your life that way is pretty important.
Thank you. So sometimes in families, you know, we get this diagnosis, and then families either, I've seen them come really close together and everybody's pitching in, and then I've seen them go away. And family members maybe don't know how to help, or really have other lives that they don't understand that maybe we need help because we don't ask. So I'd love to hear from you, Diane, about your family seems to have gathered all together. Have you seen that in some family members or not?
Mostly our family has pretty much drawn together. We get a lot of strength from the boys, and I get a lot of strength from my daughter-in-law as well when I see what she goes through. Our family has really pulled together. And I can't say that we have been distanced by this at all. Even some family members that were kind of on the edge before, I see them so supportive of everything that is done, fundraising and gatherings. They're always there. So it's been, overall, it's been very good.
Thank you. So Braden, so has Duchenne influenced the things you do in high school, the choices you make, the plans you have for the rest of your life?
Well, I think like moving forward, I've like thought about going into research in gene therapy and maybe studying it in college. And then in high school, I think that it's just kind of opened my mind up to like making sure that Micah's, or not even Micah, but like all kids that not necessarily have Duchenne, but kids that don't always have a voice in school are like heard and held accounted for. And I think that that's really shaped who I am as a person.
Thank you. So I'm going to just say Mama too.
That's fine.
Tell me about your relationship with Anthony. How did that happen?
Well, I think, and even with both boys, I kind of just, I wasn't overbearing. I kind of just sat back and waited for them to come to me. And I really don't know how it started, but Anthony and I have a thing where we call each other, he calls me a turkey and I call him a chicken. I mean, he's not a chicken by any means, but we just have, since I've met him. So that was kind of our connection. And then I don't know, the way we can talk, he has, well, when I first met him, he had no filter. You know, he would tell me all kinds of things. As he's getting older, it's a little bit more. But, I mean, just the things that we share, he's got a great taste in music. You know, I don't mind listening to his music. And just some of the, I took him for his 16th birthday to LA to see AC/DC, his favorite band. It was just me and him. So we just, I don't know, those kind of, you just have a really good connection. Yeah.
Thank you. So Colin, do you worry about your parents?
Well, that's a good question. As you know, as a son, of course, you worry about your parents, but in many ways I don't. They're great people, and I still have a lot to learn from them. Then they've been great support to me over the years. Well, my mom and I, of course, spend a lot of time together. My dad's very busy, but even when he's home, they really care about me a lot. And they'll do just about anything. They're great. So I love them dearly. I don't really know if I worry about them. Sometimes I'm sort of a little bit bummed that they need to, like just this year when we were in Austria, for my research that I was doing there, and they would see how bad it is for people with disabilities there, and they would just get so frustrated and just like, oh, why is it like this? And I would be like, you guys are my #1 fans, so of course to them it's going to seem awful, but no, I wouldn't really say I worry about them. They're great, and they're my two, well, my brother is too, but they're my two biggest fans. So.
Thank you. Braden, do you worry about your parents?
Sometimes I think maybe hopefully we won't lose anyone soon, but in the future it might bring a depression with all of us, and with me at least, I kind of shut things away until it happens. But I think that I don't worry about them. I think that in the long run, everyone will be okay.
Thank you. So Diane, if you were able to give advice to us, all of us, what would that be? What would that look like?
Wow. Look out at everybody here. I would say to find a way to put the heartbreak aside and gather all the positivity that you can, because there is a great deal to enjoy and to share with the boys. And if you look at the progression or the bad things, you're going to miss a lot along the way.
Thanks. Gretchen, if this were 10 years ago, what would you tell yourself?
Wait, a lot's going to happen. I guess mainly just to relax, make sure that you take care of yourself. Nothing awful is going to happen today. And really to make sure that I am educated and that I get the best care for Nick. I we stayed in our local clinic longer than maybe we should have. And I think now parents have more access to information because he was diagnosed pre-Facebook, pre-social media. So I would say get yourself educated, know everything, but try not to freak out, be patient, because great stuff is coming down the pike.
Mike, what would you say if this was 10 years ago?
If this was 10 years ago,
What would you tell yourself?
What would I tell myself? Well, I know 17 years ago, there was nothing like we have today, and really 10 years ago, pretty much the same thing. So kind of like what Gretchen said, I mean, you have to just, live every day. We try and keep things normal. Look at how much stuff is going on right now. Like Chris said today, gratitude. We have a lot of gratitude, for being a part of this family. And, Michael was able to get into a trial after all these years. So, that was big for us. But I am not a worrier. I'm usually telling my wife, relax. I like to worry when it happens, So, yes, it sucks. But, we try and keep things as normal as can be, but yeah, I would just say try and, don't freak out. You have to, we don't make it, Duchenne is not like we're home, we don't talk about it. We do our thing, every day. And I thought Chris's presentation was great. He said it's those in-between things, and it is, we have a great time just being home with Michael's friends, hanging out. Yeah, we have great vacations and stuff like that, but it is just the times at home just doing goofy stuff, which I'm a specialist at. So, so yeah.
So, Dee Dee, what was the biggest challenge when you walked in this door? Did you ever think, oh my God, what did I get myself into?
Walked into the conference door, or walked into? I was a little nervous about that too.
Conference, right?
But this is amazing. You guys, this whole conference is incredible. I've been truly impressed and learned so much. I don't know. I don't really think I felt when I walked in the door that, wow. What have I got myself into? I don't think I ever felt that. I think at the beginning I felt like Anthony was fragile, and I was a little, and I think there's a few people that, feel that way when they first meet a boy with Duchenne or any of that. But he let me know his limitations, and we worked around that. I understood that I don't have to treat him like he's fragile, Yeah, he let me know that for sure. But he was really good at letting me know his limitations. So I just let him guide me, But I don't think I really felt like, oh my gosh. I mean, there are moments where I feel sad, and there's moments where I feel really happy, but all in all, I don't think I ever felt overwhelmed by it. No.
Good. So Gretchen, back to you again. So tell me, if this was the beginning, right? If this was that new diagnosis for you, and you came into this room, what would you predict for a future today? I mean, you don't have to say X, Y, Z will be approved, but how would you look today if you were starting again?
When I was looking at the last polling thing, I didn't have my phone because I was right over here. I don't know if Brian answered the right way. But I like the idea of some kind of like a cocktail of drugs. We use cocktail, but Nick's been in a study where he's been taking medication 3 times a day for off and on for 12, 14 years, something like that. And for boys to have to choose or parents to have to choose what they're going to put their child on, and then have to hope that it's the right thing, and then if it's the wrong thing, they can't get into the next thing. So I would wish for or hope for something that we could have our child in something, and if they're not being negatively, adversely affected by it, that they could try another thing without having to wash out something that potentially is keeping them from getting pneumonia or from having further heart issues. And I'd like to see that, and seeing the amazing slides and how things have developed. I think there's potential for that.
So, Diane, I'm coming back to you again. Just to ask you, you quit your job, is that right, to help the family?
I did. I cut my hours when school hours changed for Kevin, because Dee used to pick him up after school. She was a teacher, and it didn't work anymore, so I cut my hours at work. And then I told them, you know, when there needs to be more time spent with the boys, that's when I will leave. And I did, and it was the best thing for me to do. It gave me so much more time to spend with them and to help out. And I've never regretted a minute of it. I miss my clients, I miss the people I worked with, I miss what I did, but there are no regrets. It was the right thing for me.
Thank you. So Braden, can you tell me if you had something to say to siblings, because I think it's hard to be a sister or brother when you're healthy and wish that you could change the world. What would the advice be that you'd give to siblings?
I'd probably just tell them, or I don't know, I've realized that when I was growing up, like, I didn't always want to do like I didn't want to like mow the lawn or like go to sports and like do certain things, but then I kind of realized that, you know, kids with Duchenne like can't do those things, like even if they want to. So I just kind of like realized in high school that I should just kind of like do it for like Micah, because he would do it if he could. So I would probably just say, whatever you do, or like whatever things that you complain about, just kind of remember that you have it easier than others. So.
Good, thank you. So Gretchen, back to you. You work full time?
I do.
Yes. And would you give advice about working at least outside the home? Does that help you, or are there days when you
I would say don't feel guilty if you do, and don't feel guilty if you don't. For me, it definitely helped me to compartmentalize and be able to separate. Nick, once he was in school, it was, because he was in daycare when he was younger, and then with his physical needs, I had to be a little more on that with people and with getting him rides to school. But it helps me put things into perspective, gives me sanity, and definitely, I can't wallow in my own life problems with my kid, because it again helps put into perspective that everyone has problems, and overall in the grand scheme, our family has it pretty good, including Nick. So.
Thanks. How many of you are on Facebook? I'm assuming everyone. Yes? Yes? Okay. How much do you rely on Facebook? So we'll start with Braden. Are you on Facebook with your friends? Do you ever talk about Duchenne?
I don't have Facebook. I have Instagram, but
of course.
Yes.
And I'm not going to be dumb and say like, well, how does that work? But okay. So talk to me about how frequently do you use Instagram?
Probably too much. Yeah, a lot.
Is this ever about your brother?
I mean, sometimes like friends will post stuff with Micah, so, and he has one too now, I think. Yeah.
Okay. Do you tweet too?
No.
Okay. I'm going to get up to speed pretty soon on that. So. Okay, so Diane, I'm assuming that you're on Facebook.
I am, and I probably go on there maybe twice a month. I'm really bad.
Is that right?
Uh-huh.
So what do you go on there for?
I will go on there when I get notifications that, you know, something's coming up with PPMD, or somebody has posted something about an upcoming fundraiser. Otherwise, I'm really not on there much.
Okay. Mike, do you follow it?
I do. At least once a day, I would say. For me, I don't do a lot of posting, but it helps me keep up with, I mean, like when we're on vacation, we'll post, and a lot of people enjoy that. And it helps us keep in touch with other family members that we may not see a lot. And a lot of the friends that we've made through these conferences, because they're all over the world. Mm-hmm. So sometimes we only get to see them maybe once a year, if that. So that's really a big thing, is really to keep the connection, see what other people are doing, you know, with their kids and things like that. So.
Do you get medical advice on Facebook?
I read some things, but I don't really, you know, it's a little, I leave that to my wife. She's the medical expert.
We'll ask her later if she gets the
Yes.
advice there. So, Colin, are you on Facebook? Or are you going to tell me Instagram or something?
Yeah, no, I'm not. Well, actually, I won't tell you that, but I do have both. I'm terrible at both. Although thanks to Annie, she always posts photos of the pack, so thank you. You've really increased my social media presence. It's true, yeah. But yeah, I loved, well, now I like to see what my fellow pack members are up to, or living with MD, that's a Facebook group, connecting with. It's been especially great for connecting with people from MDA camp, both attendants and campers, so that's been great. Other than that, I don't know, I sort of have a love-hate relationship with Facebook. I'm pretty honest, and I always think that people just posting the best things about themselves is kind of weird. But Instagram, I think I posted my 6th photo in like 3 years, so I'm pretty bad. So I'm trying to get better though, but yeah.
Gretchen?
Every time I take a picture of my kids or try to take a picture of my kids with my camera, they say, you're posting this to Facebook. I'm on it. I check it all the time. I think too much. And I have been added to a lot of Duchenne groups and DMD groups. Really, like Mike said, some of my closest friends are people that I've met through this community, and I enjoy hearing about them and connecting with them, not so much through anything specifically medical, but when something happens to you know, someone's son or you know, a friend of mine that has Duchenne, I worry, I'm concerned, but I don't get medical advice very often, and I've blocked a couple people who've given me advice.
Ooh, okay.
I do use Facebook. I don't use Instagram. I have an account, but Oliver set that up for me, so I don't really use it. I'm the same. I really don't post a lot. It's kind of just to stay connected with family and friends that are spread out everywhere. Jill does have me in a couple of groups. So I do read and kind of keep up and see what's going on with people and that type of thing. But no, as far as any type of medical advice or anything like that, no. Really good for Facebook.
So while we're on the topic of medical advice, we're going to switch to marriage. Does that like link right together? So what advice would you give stepmoms out there, Dee Dee?
What I would say is be open, put your egos aside, love. Just things don't have to look like everybody thinks they're supposed to look. You can, I mean, like I said, when Jill and I first met each other, and with Chris, we put our egos and stuff aside. Well, what we found out later on is we actually really like each other, and we have a lot of common interests. Her and I took the boys on vacation, just me and her with the boys, and had so much fun. So it's possible. Just keep that in your heart and in your mind, that it is possible.
So Gretchen, marriages are hard in general, and maybe more specifically in this environment. So can you tell me how did you keep it together?
I think we're pretty honest and open with each other. I'm pretty good at complaining, and I think if we have issues sometimes with our boys getting along. My older son Alex is exactly like my husband Brian, and so they butt heads all the time. And I think with Nick, sometimes we might disagree about things, but Brian will defer to me a lot about some care things because he knows that I'm more focused on it and stay more in touch with things with the community. And when I feel like I'm gonna lose it or I'm frustrated, when I do get upset or overwhelmed, he's usually quick to be there and be a more calming influence on me.
Can I ask you one more question about this as well?
Maybe.
Maybe, is that what you said?
Maybe I'll answer it, yeah.
Okay. Okay, so what advice would you have to give all of us, right? How do you keep it together? How do you keep from wishing murdering someone?
For parents or for wives or
for parents?
Everything. I think this morning when we were in the mom's yoga session, which I hope everybody got a chance to go to because it was awesome, that idea of making sure that you are taking care of yourself. And being a martyr doesn't look good on anybody. So if you need help with something, if you feel like you can't cope, reach out and ask somebody for help, whether it's your medical provider, if it's a spouse, if it's a sibling, if it's a friend, if it's a neighbor. Don't be afraid to ask for help. Definitely, everybody who's in this room is already aware of and familiar with PPMD, but the more friendships you cultivate here and relationships you have, I think that's very, very valuable. And I'll say it again, everybody has issues, everybody has problems, so be empathetic and watch your wallow. Don't get too caught up in your problems.
Thank you. So, Colin, and I'm going to ask all of you this. You now have adults in your house, and sometimes politically or in other areas, you have differing opinions. So how is it to live, Colin, do you ever disagree totally politically or otherwise with your parents, and how does that get resolved?
Well, no, we actually are pretty similar in a lot of respects. We both, my parents both really value, logic. They're both scientists. My dad's a physician, my mom's an engineer. So everything in our house has to be like dissected, like rationally. Sometimes I'll say funny things like, oh my gosh, I think, I don't know, I'll come up with some crazy thing like, oh, my back hurts today because, I don't know, it's raining. I would never say that, but that's an example. And my mom would be like, no, sorry. So and I think because of that, we actually end up being on the same side of a lot of issues. My brother and I are probably a little bit more liberal because we're both artists, we're both musicians, but actually, we're all sort of, because of sort of my experience, really I can speak to something different, which is, sorry not to change the question slightly, but those who have had different, political or religious views or something, and then having, encountered me, they sometimes kind of come to see things in a different way. So that's kind of been more of my experience, is that for different families, sometimes they had maybe a notion about something, and then they see, oh, well, he's got a lot of differences there, maybe I should be more open about other, other groups, for whatever reason they might be different, so.
Thank you. So Mike, how do you hold the marriage together? Just do what Suzanne wants?
Exactly. She will not say that. No, we.
Just kidding, Suzanne.
Oh, come on. No, we. I always preach this. Always played sports. We're a team, and I tell that to Michael when he starts to veer off the team, right, Mike? I said, we're a team. Why, you know, you got to come back into the team here. But we definitely try and make time for ourselves, for sure. Especially now Michael's older. He's been blessed with a lot of good friends. So we're like, who's coming over tonight? All right, we're going out. So, you know, we definitely try and make time for ourselves. You have to do it.
Yeah.
Or you're just, you know, you'll just separate. You know, it won't work. You have to have time for yourself. So.
Yeah. Thanks. Diane, what advice would you give?
What advice would I give?
Yeah, to keep the marriage together. Do you ever see things peeling apart and kind of want to step in and say, I'm going to fix
well,
this? Or?
Since I've been divorced for 28 years.
Yeah, but no, but we're not talking about that one. We're talking about the other one. You're watching. Yeah.
I try to give Mike and Dee time together. I'll be over there. They've gone on, you know, 7-8 day cruises, and I've been there hanging with the boys and getting them to school and getting them up, and it's important to do that. They also have friends who will come over and hang with the boys so they can go out. And I think it's important for them to just spend time together when they can. And they also spend, especially my son, spends time by himself, you know, off doing other things. So it's important to have that twosome time away as well.
Thanks. Braden, how do you encourage Micah to be independent?
I don't really think I need to encourage, because he's pretty independent. He spends a lot of time playing video games, so but he's also social. But I don't think I've ever had to encourage him to be independent.
Gretchen, how do you encourage independence?
I think bringing Nick to the conference helped him to see, other guys with Duchenne, tooling around and doing things. And then he turned 18, and there was always an expectation that he was going to go to college because, that's what you do. And so we never strayed from that, really, that there might have been a question of whether he would be able to live away at school, but we never discouraged that. That was sort of his own doing. We happen to have a very accessible university that's about half an hour away, and I don't really know that we did anything, but he, just finished his first year of college, and he lived in the dorms, and he was able to set up his own care for the most part. And I guess we encouraged him or supported him by letting him know we would be there if he needed anything. If it didn't, if he was having issues, he could be open with us. We do worry that he tends to stick with the friends that he already has and isn't super outgoing that way. I do think he spends too much time playing video games, but he's social in that. So in a different way of looking at it, he's able to put on his headset and talk to his friends without having to go to their houses. And he has friends in Texas that he met from playing video games and stuff. So
do you worry about him online? I mean, at all?
I don't, because he's a pretty sharp cookie, and he is most of the time he's playing with the friends that he's pretty tight with. But he's 19, and he is a pretty solid decision maker, so I don't worry about that.
Thank you. So Colin, how are you independent, and how much independence do you sort of take to make friends, go out by yourself, go off on your own?
Okay, yeah, well, I went to college about an hour away, in Hope College in Holland, Michigan. We have a Tulip Time Festival when we have a working windmill, so that's interesting. So yeah, I hired my PCAs when I was there. They were all students, wide range. Some of them were, one of them was a nursing student, he was great. A couple of them were in a fraternity, you know, that was great. And the campus was really close to the downtown area, so that was really great. I was just able to walk downtown, and you know, that was great. For, you know, grad school, I'm, you know, going to a university in my hometown, so I'm living back home, and it's a little bit harder, you know. I live further out. I live about 20 minutes from the, you know, from campus, but I've gotten the hang of using my local, you know, transit service, and that yields lots of funny stories. Buddy Cassidy can tell you all about those. He used to post about the ones he experienced in Virginia. Good stories, but anyway, so that's been great, and I use that a lot. And I'm always willing to tell my friends, you know, how to use my van, and you know, they'll come over and we'll drive out in the van and you know, show them how to use it. I would like to continue, maybe to live independently again. I'm applying for PhD programs, so one of my top ones is University of Michigan. So if I did that, I would live in Ann Arbor. So but I'm just open to other places too. So I'd like to live there, and you know, you know what the timetable is, but I'd love to teach for as many years as I can, and wherever that would be. But socializing can be tricky. I'm not really the most extroverted person ever, but I do have a good group of close friends, and yeah, so that's good.
All right, it is time to party. This conference is now officially ended, and we are going on to around the corner, I think, to the bar, and then to dinner. So I wanted to thank all of you. Next year is Orlando, Florida, June 26th to 29. I don't know where we'll be in 2020, but we may well land back here at the Phoenician. I think it's been a really wonderful conference. I think the place is beautiful. I can't thank enough all the international friends that have come to this conference and all the locals from the United States. So thank you all for joining us. And this panel is pretty terrific, smart people.